So this is unit 15.5, and I'm on page 528, and we are discussing phospholipids. So these are a category of lipids, and there are two categories in phospholipids. There are glycerophospholipids or sphingophospholipids. Sphingolipids and glycerophospholipids. So this is glycerophospholipid. You see there is a glycerol molecule connected through an ester bond to two fatty acids. So there are two bonds here. This is a bond here, and this is again a bond. So there are four bonds connecting these five different molecules together. So glycerol, fatty acid molecules two, one phosphoric acid, and one amino alcohol. So these are the components of glycerophospholipid. Again, where is our emphasis is looking at the name and setting up the structure and looking at their function. What is the function of this particular molecule? Now again, contain two fatty acids. They form ester bonds with the first two hydroxyl groups. These are the first two hydroxyl groups, and these two fatty acids are connected through an ester bond. The third hydroxyl group connects to the phosphoric acid, which forms another bond, an ester bond with the alcohol. So this is the basic setup. So there are four components. Sphingomyelin, which is the other phospholipid, contains, instead of glycerol, there is this sphingosine molecule, which has its own long chain carbon entity, but one hydroxyl group connecting to a fatty acid, the second connecting to phosphoric acid, connecting to an amino alcohol. So this is the basic structure of sphingomyelin. And what are these two? These two are phospholipids. We are still in lipids looking at phospholipids. So remember, in sphingomyelin and glycerophospholipids, we have said that one of the components is an amino alcohol. These are some amino alcohols: choline, serine, and ethanolamine. These are some of the amino alcohols, three. So now our physiology inside is a weak base. The pH is 7.4. So at that pH, these amino alcohols that are present in the lipids are ionized. You can see the ion. So these are ionized at the pH in the body. So what are these? These are one of the components in the glycerophospholipids. Now, with that, these are the two glycerophospholipids that we are looking at: lecithin and cephalin. So here are the amino alcohols, the two amino alcohols. Of the three we saw in the previous slide, choline and ethanolamine. These are the two amino alcohols. This is the phosphoric acid molecule. This is the glycerol, and these are the two fatty acid molecules. Here again, this is the next glycerophospholipid: glycerol, two fatty acids, phosphoric acid, and the amino alcohol. So these are the two glycerophospholipids. They are abundant in brain and nerve tissues. They are found in egg yolk, yeast, wheat germ. So these are the two examples. So look at the structure. Be comfortable setting up the structure. Look at where they are found and what their function is. We will get to it as we go along. So glycerophospholipids have a polar and a non-polar region that allow them to interact with polar and non-polar substances. And this is a very important property of glycerophospholipids. These have a polar head. The amino alcohol part of the glycerophospholipid is a polar end. It's called the polar head. The non-polar tail is where the fatty acids are connected. The glycerophospholipids are most abundant in cell membranes and play a very important part in cellular permeability. So this is what is the most important thing about glycerophospholipids, is that their structure of having a polar head and a polar tail is very useful in cell membrane. So we'll see how that happens, but first we are understanding the structure of glycerophospholipid, how it has a polar and a non-polar part to every molecule. So the polar is attracted to water, it's hydrophilic. The amino alcohol and the phosphate portion, remember there is a phosphoric acid, that portion is water, it's attracted to water, hydrophilic. The non-polar hydrocarbon tail is non-polar and soluble only in lipids. Now this solubility in non-polar substance as lipids is a very important point that will come later in the use of these glycerophospholipids. This is what we are talking. Again, if you cannot see this well, please refer to your book, and this is the expanded structure where we are explaining the polar head and the non-polar tail. This is the amino alcohol and the phosphoric acid part, the polar head. This is the fatty acid part, the two molecules of fatty acid, which is the tail. And when you look at it, this is a simplified version, so you can look at it as a head and a tail. So this structure is a very important structure to understand. Why we are looking at this in this way is because the function is very important. Now please refer to your book if you're unable to see the structure clearly. It is in the book. The book will be your best friend. I can tell you it's mine. So again, this is also snake venom. This picture scares me of the snake. Poisonous snake venom contains phospholipases that hydrolyze phospholipids in red blood cells. And that's again another area where you find glycerophospholipids. Okay. Identify each of the following as a fatty acid, a triglycerol, a amino alcohol, glycerophospholipid. I will look at this name, glyceryl trioleate. So I see glycerol and 3 oleic acid molecules connected. That is a triglycerol. Cephalene, choline, palmitic acid. Palmitic acid is a fatty acid. This acid. Choline is an amino alcohol. What is cephalene? Can you remember where you heard cephalene? So there, glyceryl trioleate is a triglycerol. Cephalene is a glycerophospholipid. Cephalene, and what was the other glycerophospholipid was choline. Cephalene and choline, cephalene and lecithin, sorry. Cephalene and lecithin are your glycerophospholipids. Choline is one of the amino alcohols. What are the other 2 amino alcohols? Choline, serine, and ethanolamine are your amino alcohols. So we are trying to remember all these. Lot of remembering and pronouncing them right. Draw the condensed structural formula of a cephalene that contains stearic acid and serine. Okay, what is cephalene? Is a glycerophospholipid. It contains serine as the aminophenol and stearic acid as the fatty acid. So let us see, how do you set up? What is stearic acid? I'll go back to 15.5. Page 515, and look for what is stearic acid. How many carbons? I see 18 carbon chain with no double bond. That is stearic acid. And what is serine? That is the amino. This is the amino alcohol, and I'm on page 528, and there I see a serine molecule. And now I have to set up the structure of the glycerophospholipid. There is my structure of the glycerophospholipid. There is glycerol. There is the stearic acid molecules connecting to the two hydroxyl groups with the elimination of a water molecule. Again, stearic acid, 18 carbons with no double bonds. And this hydroxyl connected to the phosphoric acid connected to the serine amino alcohol. So this is how I set up, and this is what we are trying to do. Set up the structure looking at the name. Sphingomyelin. Sphingomyelin, this is the next phospholipid. The amine group of sphingosine forms an amide bond to a fatty acid. Again, in this, I wanted to tell you, please look at page 530. Now, while we are looking at this slide, please look at page 530. And there you see the molecule, the basic structure of sphingosine. I'm looking at sphingosine, this structure on page 530, sphingosine. You can see that it contains an amino and a hydroxyl. That's what they are talking about. The amino and hydroxyl giving an amide bond and an ester bond. So where is the amide and the ester bond being formed? It is at the amino and the hydroxyl group. So sphingomyelins are abundant in the white matter of the myelin sheath, a coating surrounding the nerve cells that increases the speed of nerve cells and insulates and protects the nerve cells. So this phospholipid has a very important function, as you can see. It protects the nerve cells, it increases the speed of nerve impulses, insulates and protects the nerve cells. And it is located in the white matter of the myelin sheath. That is a coating surrounding the nerve cells. So sphingomyelin. Now, this also is on page 530, the structure. The structure of sphingomyelin containing myristic acid and choline. I don't know if this is myristic acid or palmitic acid. It says palmitic acid. Check the acid. Which acid are we talking? It says myristic and palmitic. So 14, 15, 16 carbons with no double bond. So let us see, 16 carbon with no double bond will be palmitic acid. Myristic acid is 14 carbons with no double bond. So this is palmitic acid. So this should be palmitic acid. So there is your sphingosine molecule. This is a part of the sphingosine molecule. So this is a part. The only two bonds that we are seeing is an amide bond with the ammonia in the sphingosine molecule connecting to the acid, a bond, hydroxyl bond with the phosphoric acid, and the phosphoric acid forming an amino ester with the amino alcohol. So 1, 2, 3 bonds. This is a part of the sphingosine molecule. So setting up a sphingomyelin molecule. So you will be given the acid. What will you be given? The acid and the amino alcohol. This is a permanent factor. This phosphoric acid molecule is always there in phospholipids. In the name, phospho. Palmitic acid, a 16 carbon saturated fatty acid, is the most common fatty acid found with the ionized amino alcohol choline. Draw the condensed structural formula for the sphingomyelin. So we'll start with the sphingosine molecule. And this is fatty acid most common, 16 structure saturated palmitic acid. So I would ask you to look on page 531, and there is your condensed structure, 531.